Hi everybody, this is Vasaki from Chandu Biology classes. So the most awaited notification has been released by the government of Andhra Pradesh for JL and DL exam. As per the notification, the vacancies to be filled under JL is only one for both botany and zoology. With respect to this DL, the number of vacancies are 19. 19 for botany and 19 for zoology. So by considering all these difficulties, Chandu Biology classes has decided to conduct a class for botany and zoology. So what we can ensure you by joining our classes are complete completion of syllabus. 100% completion of syllabus will be guaranteed. And the second one is complete analysis of previous year question papers. And the third one is complete notes for each and every topic which is mentioned in the syllabus will be given to you. And the fourth one is after completing each unit, unit-wise test will be conducted. And the most important thing is doubt clearance will be available in no specific time. So the courses will be started from 11th Jan. You can make use of it. Now in this session, I'm going to discuss about the analysis of syllabus of zoology, particularly 4 units. Okay, in zoology there are 11 units totally. Uh, uh, In that 11 units, general concepts, core data, and histology. These 4 units will be handled by me. Remaining units will be discussed by Mr. Chandrasekhar sir. Now let us focus these 4 units. Okay. Whatever the exam is, the first thing an aspirant can do is knowing the syllabus. You should aware about each and every word given in the syllabus. This is the first thing you should do. Because the syllabus is the direct voice of the committee who is involving in the preparation of the question paper. And after knowing the syllabus, you should know at least 10 to 15 points of each word given in the syllabus. Let me explain in the detail. See, first, general concepts have been given. In that general concepts, the first thing is a level of structural organization. In this, you want to read about the classification of organisms. Why classifying organisms is very important and on what basis classification has been classified. In that, many subtopics have been given, unicellular, colonial, unicellular, colonial, and multicellular forms. In unicellular, you should know what is unicellular and which phylum will be placed under this unicellular. Okay, and colonial, what is colonial, which organisms will exhibit in colonial form and which organism will exhibit in solitary or individual form. Like in the same way, what is multicellular and which phylum is placed under the multicellular forms. Also, you should know what is prokaryotic and eukaryotic cells and what is its difference, the basic difference between prokaryotic and eukaryotic cells to be understood. And the next one is level of organization of tissues, organs, and system. Here you want to read what are all the grades of organization. Generally, there are 4 grades of organization, which is cellular level of organization, tissue level of organization, organ level of organization, and organ system level of organization. We should not stop with that. Why a plant is placed under the organ level of organization? Why animal is upgraded to the organ system level of organization? This understand is very much important. Why fungi is said to be as the tissue level of organization? We should know the basic difference between this grade of organization. Now next, acoelomates. Acoelomates, pseudocoelomates, coelomates. See, what is acoelomates? What is pseudocoelomates? What is coelomates? Generally, coelomate means body cavity. Here, acoelomates means absence of body cavity. Pseudocoelomates means false body cavity or fake body cavity. Coelomates means true body cavity will be developed in these organisms. Here you should know that what are all the phylums which is placed under this acoelomates what are all the phylums placed under this pseudocoelomates and what are all the phylums which is placed under the coelomate. This is very much important. And you also want to know that these acoelomate and pseudocoelomate coelomates, they are the subdivision which is placed under the division Proterostomia and Deuterostomia. This is very much important. Okay. For example, acoelomate. What is acoelomate means? There is no body cavity has been developed in that particular organisms. And what are all the phylums which is placed under the acoelomates? That you have to read. And this acoelomate is one of the subdivision which is placed under the division Proterostomia. And now, what is pseudocoelomates? Pseudocoelomate means false body cavity. It looks like a body cavity, but actually it is not a well-developed body cavity. They are said to be a pseudocoelomates. And what are the phylums which is placed under pseudocoelomates? Like in the same way, coelomates. What is coelomate means? A well-developed body cavity is developed in these organisms. And what are all the phylums which is placed under these coelomates? And what are all the types of coelomates are there? Generally, there are two types of coelomate. One is isocoelomate and enterocoelomate. That enterocoelomate which is placed under the division Deuterostomia. This basic level of understanding is very much important. If you know these understanding, you can easily answer the question. Now, coming to the third division, concept of species, hierarchical strata, binomial nomenclature, classical methods of taxonomy of animals. What you should know in this line is you should know the hierarchy of the taxonomical classification. When it comes to the hierarchy of the taxonomical classification, the top place have been held by the domain. Domain is nothing but either it is an eukaryote or prokaryote. That is considered as a domain. In that domain, many kingdoms are present. That is plant kingdom and animal kingdom. In that kingdom, many phylums are present. Phylum, there are generally 9 types of phylums, starting from the protozoa and end with echinodermata. All these are comes under the phylum. After the phylum, each phylum has many classes. Each classes have many orders. Each orders has many family. Each family has many genus. Each genus has many species. This is the hierarchy of the taxonomical classification. This you want to know. These are all the basic general concepts before a non-core data. See, all the phylums, all the 9 phylums are said to be as the non-core data. Okay, next. Um, before we get into the non-core data and core data, we should know the basic difference between what is non-core data and core data. What are all its differences? And difference between invertebrates and non-chordates, and difference between notochord and vertebral column. You should know what are all the meaning of these terminologies. So after that, you should come to non-chordata. In non-chordata, we know there are 9 phylums: Protozoa, Porifera, Coelenterata, Helminthes. In Helminthes, there are 2 phylums. 2 phylums are generally said to be as Helminthes. One is Platyhelminthes, that is flatworms, and Nemathelminthes, that is roundworm. These 2 phylum are said to be as Helminthes. Annelids. The name annelids is generally considered as a segmented worm. That is, it shows the metamerism. And Arthropoda, Mollusca, and Echinodermata. First, after you should know that after you have read the difference between these non-chordata and chordata, invertebrates and non-chordata, notochord and vertebral column, you should read the general characteristics and classification of all the phylums. Like starting from the protozoa and ends with echinodermata, you should read the general characters and classification. That is the most important thing. If you know the general characteristics of protozoa, you can also able to understand why amoeba is placed under the protozoa. And if you know the general characteristics of Coelenterata, you also come into some understanding why corals are placed under the Coelenterata. If you know the general characteristics of echinodermata, you also can easily understand why starfish is placed under the echinodermata. So each phylum has its unique features. Based on these features, organisms are placed under the phylum. Right? Fine. If you read the general characteristics, then whatever the question may be, you can easily locate the organisms in the phylum based on the characteristics itself. After reading those general characteristics and classification, coming to the subtopics given in each phylum. That is protozoa. In protozoa, they have given locomotion, nutrition, reproduction, and protozoan diseases. First, let's take locomotion. In locomotion, you should know that what is locomotion and what are all the locomotory organelles used by those organisms which is placed under the protozoa, and what are all the special features of the particular organelles, and what are all the types of locomotion seen in the protozoan. Generally, there are different types of locomotion seen in protozoa, like ciliary movement is there, amoeboid movement is there, flagellar movement, paddle stroke movement, metabolic movement. All these types of movement will be discussed in detail here. And in nutrition, there are different types of nutrition followed by the organisms which is placed under the protozoa, like holozoic nutrition, holotropic nutrition, mixotropic nutrition, parasitic nutrition, and the saprophytic nutrition. And you also know which organisms will follow which type of nutrition. That is very important. Examples, when it comes to non-clear data, examples plays an important role. That will be the questionable area. And reproduction in protozoa. In case of reproduction in protozoa, there are different types of reproduction seen in the protozoa, like binary fission, multiple fission, regeneration, and which organisms will perform which type of reproduction. That is also important to know. And some of the protozoan diseases also discussed, like malaria, entamoeba histolytica, trypanosoma gamians. And when we discuss about those pathogen, we focus on the diseases, causative agent or pathogen, and what are all the larval stages undergo by those pathogen, and what are all the hosts needed for those pathogen to complete their life cycle. That is also important. And what are all the diseases are going to caused by those pathogen, and treatment and prevention. That is also an important thing. Next, porifera. In porifera, they have given a canal system in porifera. In canal system, what you are going to deal with this is what is canal system, parts of canal system, types of canal system, structure of canal system. All these will be discussed in detail. In skeleton in porifera, we are going to focus on the spicules and spongin fibers. In case of spicules, we are going to focus on structure and its types. And in spongin fibers, we are going to focus on the structure and its function. In reproduction in sponges, we are going to focus in both asexual reproduction and the sexual reproduction. In asexual reproduction, budding is there, fission, reduction bodies, and gemmules. They are comes under the asexual reproduction. In case of sexual reproduction, we are going to focus it has been fertilized, how the development is there. In case of fertilization, whether it is a in case of development, whether it is a direct development or indirect development. In case it is a indirect development, what are all the larval stages they are undergoing, and which larval stages finally transformed into an adult. That is all the things we are going to discuss in the reproduction in sponges. And coelenterata. In coelenterata, they have given the polymorphism. We are going to study what is polymorphism and what are all the different forms of polymorphism and what are all the functions of each forms. And most importantly, what is polypermedusa and what are all its forms. And also metagenesis. We are going to discuss what is metagenesis and what is alternation of generation and some of its important functions, why it has to perform metagenesis. And next one is coral formation, the most important area where the question has been arised frequently. What is coral and how it will be formed, and what are all the stages of coral formation. Everything will be discussed in detail during the class time. And helminthes, as I said before, there are two phylums are said to be as helminthes. One is platyhelminthes and another one is as helminthes or nemathelminthes. Helminthes is exclusively parasitic. They cause only, they are pathogen to a human. They have given some of the diseases, some of the pathogen which is going to give some diseases to human like Tinea solium, Schistosoma species, Ascaris, Ancylostoma, Ossuaris, Loa, Trichinella, Strongyloids. Whatever be the pathogen they have given, we are going to focus mainly on the common name of the pathogen. After the common name, mode of transmission, how they have been transmitted, and what are all its host, which host they are preferring to complete their life cycle, and what are all the larval stages undergoing by the pathogen, and what are all the treatment and prevention are available. Let's take an example. For example, Tinea solium. What is the common name of Tinea solium? For Tinea solium, the common name is pork tapeworm. Pork tapeworm, and we are also going to discuss the life cycle of the Tinea solium. In that life cycle, how the development has been arised. Both direct and indirect development. In case it is an indirect development, what are all its larval stages, and which larval stages will cause infection to the host, whether the host is primary host or secondary host, also what is primary host and what is secondary host, which larval stage is going to infect the host, everything will be discussed here. And also when it affecting the host, what are all the adaptations they are undergoing, everything will be discussed. And annelids. In annelida, they have been given expiratory system, coelom formation, coelom and coelomoducts. There are different class, different order, different family is placed under the annelids. Every organism will use different excretory system like nephridia, nephrostome, and nephridiophore. We are going to discuss about that. And the annelida is known for its metameric segmentation. The metameric segmentation starts from annelida. The most important thing, metameric segmentation. This metameric segmentation will be discussed elaborately when I teach the coelom formation, coelom and coelomoducts. And next, arthropoda. In arthropoda, they have given mouth parts of insects. The most important thing, different types of mouth parts have been followed by different types of insects. The types of mouth parts like chewing and lapping, sponging type, siphoning type, piercing and sucking type. Also, we also want to know what are the types of mouth parts and which insects follow which type of mouth parts. Examples are very much important here. And after discussing the mouth parts of insects, list of useful and harmful insects. That is also an important. And metamorphosis in insects. This is the most important questionable area. In case of metamorphosis, there are different types of metamorphosis followed by the insects. The first one is ametabolous metamorphosis are there. Next, hemimetabolous, paurometabolous, holometabolous, and these are the metamorphosis types, and these types also has another name. That another name is also important, and examples are also an important thing. And next, apiculture and sericulture. They are going to dealing with culturing of honeybees and culturing of silkworm, and how they are harvesting the honey and harvesting the silk. Everything will be discussed there. And the most important area is crustacean larva in arthropoda. The most important topic among the other topic in arthropoda is crustacean larva. Like what are all the larval stages seen in the crustacean means, Nauplius larva, Metanauplius, Protozoa. This protozoa is upgraded to zoea larva, and after the zoea larva, they are converted into cypress, cypress to mysis, mysis to megalopa, megalopa to phylosome, phylosome finally converted to alyma. Everything is very much important, and this chronological order of these larvae is very much important. And next, mollusca. In mollusca, respiration is there, torsion and detorsion, pearl formation, pearl industry. Among all these things, torsion and detorsion is very, very important when it comes to mollusca. In torsion and detorsion, we are going to discuss what is torsion and what is the difference between torsion and coiling and how the animal will look before torsion and after torsion and what is the significance of torsion and what is detorsion. Everything will be discussed here. Also, we are going to discuss about pearl culture. And final phylum in the non-chordata is Echinodermata. Here, echinoderm larva, as I said before, larval stages are very much important. In case of echinoderm larva, there are bipinnaria larva is there, auricularia larva, echinopudus, ophiopudus, branchiolaria. These are all there are some of more larva are present in echinoderm larva. These are all important larva, and the question will be arise from here. And another important unique feature which is present only in the echinodermata is a water vascular system. I'm going to explain you what is water vascular system and what are all the important things, important parts of water vascular system and what are all the functions of the water vascular system. Everything will be discussed there. Next, non-chordata is over. Now chordata. As I said, chordata, introduction of chordata. You should know what is vertebrates, what are all this is the phylum chordata. There are subphylum is there, subclass is there, class is there. Everything is important when it comes to chordata. I'm going to discuss the introduction about chordates, some of the introduction about chordates, their origin and their evolutionary evidence that how chordates are evolved from the non-chordates. Which phylum or which organism will be the evolutionary link between the chordates and the non-chordates. And there are also a retrogressive metamorphosis. What is a retrogressive metamorphosis? And this retrogressive metamorphosis is followed by which phylum? Everything will be discussed. And in class Pisces, it is here given that common edible fishes of Andhra Pradesh. Here they are not focusing our nation, that is India. They are focusing only Andhra Pradesh. So it is very much easy to remember the common fishes present only in the Andhra Pradesh. That is edible in nature. In amphibia, they have given pedogenesis and neoteny. The most important thing, you should know the difference between this pedogenesis and neoteny, and you should also know this pedogenesis is the process which is followed by the organisms, which is placed only under the amphibia. Also, important snakes of India and dinosaurs. These are all the factual information. You have to memory the list of snakes, which is poisonous and non-poisonous. How we can differentiate whether it is a poisonous snake or it is a non-poisonous snake. Everything will be important. And when it comes to the dinosaurs, I'm going to explain some of the evolutionary history about the dinosaurs. Next, they have given the flight adaptations and migration in birds. With respect to birds, they have come under the eighth category. Birds, the unique feature for birds is flight adaptation. So what are all the flight adaptation is there? For that adaptation, how their muscles are adapted for flight, everything will be discussed there. And archaeopteryx, with respect to this living fossil, is said to be as a fossil. We're going to discuss about this Archaeopteryx, and this Archaeopteryx is the evolutionary link between which phylum. Everything will be discussed there. And adaptive radiation and dentition in mammals. Here, there are different types of adaptive radiations in mammals have been seen there, like arboreal climbing is there, fossorial and burrowing, arboreal hanging is there, and not only the adaptive radiations, we also going to focus which adaptation is followed by which animal. That is the questionable area. And when it comes to the dentition in mammals, what is dentition? I'm going to focus what is dentition and teeth. What is the difference between this teeth and dentition? What is the function of this dentition and its significance, its differentiation of teeth, and what are all the attachments of teeth? What are all the succession of teeth? What are the dental formula? Dental formula plays a crucial role when it comes to the questionable area. So I am going to give some of the dental formula for some of the animals. That's all about the core data. And the remaining part is histology. Histology, we know that histology is going to deal with the tissue studies, study of tissues. From which tissue, which organ have been developed. The one single table which explain everything about the tissues. I'm going to provide you in the class. You can read there. There are epithelial tissue is there. There are different types of epithelial tissue there. Connective tissue, blood comes under which type of connective tissue. There are two different types of connective tissue. Dense connective tissue and loose connective tissue is there. Blood is comes under which type of connective tissue. Everything will be discussed there. Bone, and what is the difference between bone and cartilage? What is the difference between tendons and ligaments? From which tissue, which these skin, stomach, intestine, liver, pancreas, these are the organs. We want to know from which tissue these organs have been developed. Everything will be dealt in the histology. That's all about the syllabus analysis. And one more thing I want to show is some of the PYQ questions. Why I'm stressing that syllabus is very much important before knowing all the contents given in the syllabus is, let me prove it, how the syllabus, remembering the syllabus will help you in answering the question is by, let me prove to you by showing the questions. See, choose the correct pair of animal phyla that exhibit triploblastic and coelomate conditions and metameric segmentation from the following. See, consider you didn't read about anything about this metameric segmentation, coelomate condition. You didn't know anything about it, but you studied the syllabus. While explaining the syllabus, I have explained that Annelida is known for its metameric segmentation. And the segmentation will start only from the Annelida. We didn't even say a single word like segmentation or metamerism, everything before Annelida. That the concept of metameric segmentation arises only after the Annelida. By knowing the syllabus and knowing some of the important characteristic features of each phylum, we can easily answer this question without knowing the content. Metameric segmentation, we know Annelida is the one. And before Annelida, we didn't even use a word like segmentation, like metamerism, like that. So platyhelminthes will not be the answer. Obviously, Annelida and Arthropoda will be the answer. And next one is not a feature of protostome. I have explained what is protostome and deuterostome, and here in this question, not a feature. They have asked not a feature of protostome. So we can, you didn't imagine, you didn't know, you didn't read about a single word in the book or in the text which you have about a protostome, but you know the syllabus and some of the important characteristics regarding the protostome. By knowing this, you can easily, even if you didn't know the option, you didn't know during development cleavage is spiral, you didn't know what is the developmental type, what is the cleavage type. By knowing whether it is invertebrate or chordate, by knowing this, we can easily come into an answer, come into the conclusion. See, we studied protosome as an invertebrate characteristics. So obviously it is not the characteristics of chordate. So this will be the wrong answer. So C is present here and here. We know that it is completely a characteristics of non-chordate. So we can easily eliminate this. So obviously this is not the characteristics of chordate, and during development the cleavage is radial. This is the wrong answer. So this is why I'm stressing why syllabus is very much important to you. Okay. I hope this session might helpful to you. Thank you so much.